Hi everyone, in this video we are going to learn about difference between species and strain in bacteria. So biological species concept, which is applied for animals, where we define species as a group of organisms which reproduce among themselves to produce fertile offsprings. So when we define species with respect to animals, there we focus majorly on the allelic transfer between the organisms in a population. So population where the allele exchange is possible between organisms, we call or we group all those organisms as a one species, not just exchange of alleles between the organisms, but also they should have a offsprings that should be fertile. So whereas in bacteria, there the species concept is different, because bacteria, they reproduce asexually. There is no sexual reproduction for the exchange of the gametes. Therefore, that a biological species concept cannot be applied for bacteria. Then how we define species in bacteria? So to classify organisms, or to distinguish the species, or to create a organisms, or to group organisms as a species. So the major aspect in bacteria is a 16s rRNA. So 16s RNA is present in the smaller subunit of the ribosome. We know that ribosomes, they function as a their function is with respect to protein synthesis. We know that they translate the mRNA to produce the proteins. So the smaller subunit of the ribosomes have the 16s rRNA. So when a sequence is done for this 16s rRNA of bacteria, see, we in 16s rRNA two regions are present. One is a constant region and another one is a variable region. This constant region, so among all the bacteria, this constant region, the nucleotide sequence is 9 to 100% is similar, because this constant region is very essential for the ribosome function. Therefore, the evolution cannot make changes in this region. If evolution can make changes in this region, so those bacteria cannot survive. So therefore, this constant region is a very common among all the bacteria with respect to their sequence. Whereas the variable region, when we look into the variable region, so the variable region where the nucleotide sequences are not same. So this variable region, when we compare to bacteria, so the total length of this ribosomal 16s rRNA is 1540 nucleotides only. Just by comparing 1540 nucleotides, we cannot say that the two organisms, bacteria, are similar to each other, and so that we can group them as a one species. Okay, to group organisms into one species, so first we look into the 16s rRNA. So when the constant region is not required to the constant region is it doesn't play any role, or it doesn't decide whether two bacteria are same or not. Whereas when we look into the variable region of this 1540 nucleotides, the variable region definitely differs from one bacteria to other bacteria. So when we look into the variable region, and if 97% of the nucleotides in this 1540 this out of this 1540, so 97% of nucleotides are similar between the two bacteria, then we can group this we can group the two bacteria into one category. But we cannot decide just by looking into the just ribosomal RNA. We cannot decide that two bacteria is one species, because genomic DNA also is very important in deciding whether two bacteria, they fall under one species or not. So for example, if the ribosomal variable region has 97% of similarity, and genomic region doesn't have similarity between the two bacteria, still we cannot group this into one species. Therefore, the genomic DNA also is observing the genomic DNA is also very important to group the two bacteria into one species. So when we look into the that genomic DNA of the bacteria, so their average nucleotide identity between the two bacteria is very important, that we that we say that ANI, average nucleotide identity. So when we look into the average nucleotide identity, suppose this average nucleotide identity again that decides how whether we can group the two bacteria, whether we can group the two bacteria into one species or not. So that's why the first we have to look into the first we have to observe the 16s rRNA region. After that we have to look the genomic DNA region to group the bacteria into one species. And we know that in bacteria there is another a term that is called strain. So what is the strain? So strain is like a subspecies. So we know that in a species, if suppose some subspecies are there, so we call so again in a species, suppose we have some groups, like there is some genetical variation between the species itself, so we call them as a subspecies. Similarly, in a bacteria, when so when we can create some groups within a species, so we call them as a strains. So these strains they differ in some the sequence of their DNA, and also the type of the growth medium which is required for their survival is different. Also the temperature is different. So their pH values required for their growth are different. So such differences can be seen between these strains. So that's why further species can be divided into the strains. Okay, we usually classify the bacteria based on their shapes. Also we know that E. coli, for example, which is a rod shape bacteria. So but in different species we find the rod shape bacteria. So doesn't mean that all species they can have a similar type of shape. No, within the shape within a species itself we find different shapes, and between the species also we can find various sizes and shapes and all those things. So the physical characteristics are seen different within the species itself, and also between the species also. So this is about the bacteria, this biological species concept, and the concept of the species in bacteria. Thank you.